Hello everyone, welcome back to the Bio Decoder Channel. Today we are learning how to perform a t-test in R using a real wheat dataset. It would be a kind of step-by-step guide to see how to run and then how to visualize this t-test in R. So the data which I'm gonna use is a wheat data set where I have different varieties and they were exposed, they were either fertilized, they were supplied with different fertilizers, and one set was kept control. And we measured many traits like grain yield, plant height, thousand weight, and many. So in this video, we will focus on grain yield and we'll see if the fertilizer significantly increases yield. So let me give you a brief intro about t-test. A t-test is a statistical method which is used to compare the means of two groups. It helps us actually to check if the difference between group is real or just due to either that difference is by chance. So we have a null hypothesis in a t-test or z-test or ANOVA. We always use two types of hypotheses. So one is called null hypothesis and the other is called alternative hypothesis. Null hypothesis is denoted by H0 and alternative hypothesis is denoted by H1. So in our current video or in our current data set, our null hypothesis is that the mean grain yield of fertilized plants equals the mean of control plants. So they both are equal. And the alternative hypothesis is their mean values or their mean yield of fertilized plants and non-fertilized or control plants are different or they are non-zero. So first thing is you need to load your data set. So you need to write a command to load CSV and then you have to check first few rows and columns of your data set by writing head command. This helps us understand the structure of our data before analysis. Next, we run a two sample t-test using t.test function. This compares grain yield between controlled and fertilized plants. And the result will be stored in t result. So there is a column or there is a file name is t result. So next you need to print or just write t results and it will give us the t values, degree of freedom, p values, and confidence interval and the group means. Let's decode our results now. So here we got t value of minus 9.09, which means that control yield is lower than like minus 9.09 degree lower than the fertilized plants. Degree of freedom is about 58. Here you can see the p value is very small, so we reject our null hypothesis. 95 confidence interval is minus 13.87 to 8.87, showing that fertilized plants, it means that fertilized plants have higher yield compared to the non-fertilized. Next, we extract the t values and the degree of freedom from the t result. We set the significance threshold to 0.05. Then we calculate the critical t values like using qt function in R. This number will help us create a plot. It will show the acceptance and rejection regions in our plot. I created a script which you can just simply copy and paste in your R and then it will create a graph for you. Then finally, this all will result in a plot with different regions. The blue curve in the is the t distribution in this plot. So we got a plot here. And in the blue curve is the T distribution. The pink areas are the rejection regions. The red dotted line shows the critical T values. The green line is our observed T value. Since the green line is inside the pink region, we reject the null hypothesis. This confirms that the fertilizers significantly increases the green yield. So to summarize, we used a real wheat dataset to perform a t-test in R. We checked the p-values, confidence interval, and visualized the rejection regions. So if you enjoyed this video, please like, subscribe, and hit the bell icon for more tutorials on the statistics and biology. Thank you for watching the Bio Decoder channel.